Good evening, everyone. Welcome to the first online class for the subject plant physiology. This is a subject that students that are majoring in agronomy, agriculture or viticulture and winemaking should be doing as part of their Bachelor of Agriculture and Technology. Here we have lecture one. This is an introduction to the subject plant physiology. Now, I won't be going through this lecture because a PDF is available on Moodle. Also, I'll show you where to find the links to YouTube video recordings of this lecture and subsequent lectures a little bit later in this class. Dr. Nikki Cooley is the subject coordinator and one of the lecturers in this subject, and here are her contact details. My name is Silvana O'Coney. I'm part of the teaching team in this subject with Nikki. Here are my contact details: email, office at the Epping campus, Building A, Room 1A130A, and my office phone. If you do call and I'm not there, I'm most likely teaching, so please leave a message with your best contact number. Now, I'd like to look at certain sections of what's called the student subject outline. For new students to the course, you will receive a copy of a subject outline for each subject that you're enrolled in. These can be found on Moodle for each subject. This document's also available on Moodle, but I'd like to just focus now on the requirements for this subject, given that plant physiology is only delivered by blended or online delivery, apart from a 2-day prac workshop, which I'll come to in just a moment. So it's important that you know what's expected of you in terms of assessment, and here is the assessment information that you'll find in this document. There are 3 assessments for this subject. Let's look at them in turn. Assessment number 1 requires you to write 2 lab reports, but they're not your typical formal scientific lab reports. Let's look at them in turn. Report number 1 requires you to complete a prac booklet based on do-it-yourself or DIY practicals that you will conduct at home once you've received what's called the DIY kit. So you'll complete this DIY prac booklet and you will submit that in the last week of semester, week 13, 24th of May by 12 p.m. Report number 2 also involves you completing a booklet, a prac booklet, but this time this is based on a 2-day prac workshop, which will be conducted at the Epping campus. Here are the dates here: Thursday and Friday, 10th and 11th of May. So make sure you set those 2 days aside to be on campus to complete the prac workshop. Now, the prac workshop booklet will be completed mostly within those 2 days. There will be some bits and pieces that you might have to do at home in time to submit that booklet the following week, week 12 rather, the 17th of May by 12 midnight. Assessment number 2 is a plant physiological management plan. I'll come back to that a little bit later on. The word limit is 1,200 words, not including your references. This is worth 30% of your total mark for the subject, and it's due in week 10, 3rd of May. And by the way, let's go back to the prac reports. Each of those prac booklets is worth 15%. Finally, a written exam. This is a final exam conducted in the end of semester final exam period. The exam timetable is normally released about the middle of semester, and I'll let you know as soon as that's the case. This is a 2-hour exam, which is based on the entire lecture series for the subject. Final exam is worth 40%, and here is the 3-week final exam period. Normally our exams are scheduled within the first week of this exam period, but as I said, I'll let you know when the exam timetable has been released. Okay, you'll see something like this in each of your subject outlines. It's essentially a schedule for the entire semester, and for the new students, our semesters are divided into 13 weeks, and we're here in week 1 with our Introduction to Plant Physiology lecture, which I just briefly showed you earlier. Is our first Zoom classroom. And there is another lecture which you'll be required to study in the first week, called Root Structure and Function. All right, in week 2, we will also have a Zoom online class at the same time, 6:30 p.m. And then thereafter, the online classes will take place every 2 weeks. As you can see, according to this schedule. So we proceed through the first 6 weeks of the semester, and then you'll have a mid-semester break, 1 week off. Now, this is a break from attending class, but it should not be considered a break from your actual studies. So use this time effectively to be working on assessments, catching up with lectures, and so on. Then we move into week 7. So you'll note that the mid-semester break is not given a week number. Week 7, and we proceed through to the final week of semester, shown here, week 13. Now let's just go back to week 11, because this is when the 2-day prac intensive workshop will be held, as I said, on the Thursday and Friday of that week. And the prac workshop will run from 9 a.m. until 5 p.m. You'll also note here in week 13, there's an additional workshop, workshop 2. This is a 1-day classroom-based workshop. This is optional. You don't have to attend this workshop, although it's very useful if you need some extra help with the important process of photosynthesis. This will run from 9:30 until 3:30 approximately, and as well as doing some modelling to help in your understanding of photosynthesis, there'll also be some revision on the day. So remember, that's an optional 1-day classroom-based workshop. You'll notice in this schedule of semester that the assessments are shown here. The due dates for the assessments are shown there. Okay, when semester's finished, then we have a 1-week study vacation. After that, we enter the 3-week exam period. Okay, now let's talk a little bit more about the Plant Physiological Management Plan assessment, which I mentioned earlier. Now, before you can start this assessment, you'll need to work your way through the first 10 or so lectures. So Nikki recommends that you try to get through those lectures in about the first 6 weeks. In that way, you'll have a good understanding of the major physiological processes that occur within plants. But also, before you start on this assessment, you'll need to be allocated a specific crop plant or other plant species. All right, the crops or plant species available for selection are listed here in this table. You can see there's quite a selection here. What you need to do is look at these and select your 3 preferred plant species in order of preference from 1 to 3. Email your choices to Nikki Cooley, the subject coordinator, and she will let you know which one is allocated to you. And this is because every student has to be working on a different plant species. So don't forget to have a look at this and email Nikki, preferably in the next week or so, with your 3 choices. All right, let's move on now to another one of your assessments, the DIY prac. So this is the DIY prac manual, which is available on the Moodle site for this subject. And this will explain in detail the experiments that you're required to undertake at home. And I would just like to go through the pracs, which are listed here. So there are 3 pracs that you're required to conduct. Prac number 1 looks at plant growth and development, and there's several activities within each of these pracs that you're required to complete. Prac number 2 is looking at structure and function. So this is essentially the fundamentals of plant anatomy. And then prac number 3, a transpiration, very simple, short transpiration experiment. All right, here we have some of the items that you'll find within the DIY prac kit once you receive this in the mail. Now, most students have actually provided their current postal address, so they've either received their DIY prac kits or they should receive them later on in week 2. If you still haven't provided your current address, please do so as soon as possible. Email this to myself or Nikki or both. So what you should receive in these DIY prac kits are seeds. This year you'll be receiving oat seeds. You'll be receiving plastic pots or containers. You will need to sow 5 seeds per container. You will also receive the potting mix and some slow release fertiliser. Plant labels. You will receive a mini DIY microscope and a set of microscope slides. All right, rather than going through the instructions here for completing the pracs, I'd like to look at what you're required to do. And that is, you need to complete this DIY prac report answer booklet, also available on Moodle. So let's run quickly through all the tasks. And you'll actually note that a lot of these activities you can complete before you even receive the DIY prac kit in the mail. So the 3 major pracs, once again, let's start with the first one. Prac 1, plant growth and development. So you will sow your oat seeds and grow your plants over an 8-week period, during which time you will take some observations and record those observations in the tables shown here. So your initial observation will be plant observation number 1, the date, germination. So how many seeds germinated? For example, pot 1, 3 seed, pot 2, 4 seed, pot 3, 3 seed. Plant height in centimetres, plant height, the mean of your plant height measurements and standard deviation. Tiller number, obviously you won't be recording tiller number when during your first observation because the tillers won't have developed, but when you do, later on you will also calculate tiller number mean and standard deviation. You'll also be recording the developmental stage of your plants, and this is measured via what's called the decimal growth scale or Zadox scale. And once you work out how to use this, you will come up with what's called a growth score. In the DIY Prac booklet, you'll be given a link to a website that explains exactly how to determine the growth score. And finally, mean and standard deviation of your growth score. So that's observation number 1. Then 1 week after the initial measurement, you will take down the same observations as part of observation number 2. Then we go to the next table. Observation number 3, which is 2 weeks after your first observation, 3 weeks after in this column. Next table, 4 weeks after, 5 weeks post-germination, 6 weeks and 7 weeks post-germination in this table. So the same observations will be taken here. Okay, the next part of this prac requires that you insert a graph, a scatter graph that illustrates plant height changes over that period of time. In the next box here, you'll insert a photo of your plants, and this is to evidence that you are in fact growing those plants, and that's where your observations are coming from. And in task 3, still in practical 1, you'll be required to link plant growth and developmental concepts with the observations that you make. So you'll be answering some questions which can be answered here. And that's the end of prac 1. Prac 2, plant structure and function, plant anatomy. Also, you'll be doing just a little bit of plant classification here. So looking at figure 1 and doing a little bit of extra reading, you'll be required to fill in this table. Your next task is to label this image here. And by doing so, you'll be able to complete table 2. So this is quite simple. Most of you will remember many of these concepts from your first year in Living Systems. Task number 3, label this cross-section of a leaf. And to do that, you complete table 3. Label this image of plant cell by completing table 4. And finally, very simple, what is the function of the following plant cell structures? Another task, again, this relates to living systems, an image of a flower, and you need to label as indicated there and complete table 5. Okay, part 2 and still on prac 2. Now for this part of prac 2, you will have to use your DIY microscope slides and mini microscope. You will prepare a low power diagram of the transverse section that you see of a dicot root on one of your provided slides. You'll insert your image here and label it accordingly. Okay, this is the same sort of exercise here, but now you're looking at a transverse section of a ZMA's primary root. Same thing again, but now we're looking at a stem, a cross-section of a ranunculus stem. And another example of a stem, but this is a woody stem. So some of your labels will be quite different to the previous one. Here you'll look at a section of a monocot leaf and labelled accurately. and a transverse section of a dicot leaf. Answer one question, and then look closely at one of the leaves from your growing plants. Draw the leaf and label the vernation pattern that you see. Quite simple. And that brings us to Prac 3, which is a very simple look at plant transpiration. And to set this up, you'll have to use one of your more mature pots of oat plants and complete the tables as indicated. Finally, answer some questions, and that's the end of the DIY pracs. Okay, our final important document, which I'd like you to read through. Again, this is found on Moodle. Why study plant physiology? So within this document, you'll find links to YouTube video recordings of the first 10 lectures for this subject, which represents the first 10 weeks of semester. Now here you can actually proceed to work your way through these lectures, taking notes as you go, and you'll notice when you actually start with these lectures that often there are links during the lectures to YouTube video clips or additional readings and so on. Now, if you're anything like me, I would find that, and I do find that, a little bit distracting from the lecture itself. So I would probably ignore those references to links and additional readings until I got to the end of the lecture, and then go back and follow those links or do the extra readings. So that's it for week 1 of plant physiology. Our next class will be next Thursday evening at 6:30. Remember, if you've got any questions at all, please let me know by email or by phone. Have a good week and good night.